Hello, college biology. This is part 2 of our fluid mosaic model videos, and this video will deal with the mosaic component. The mosaic means a lot of different pieces coming together, and that idea just comes from the fact that the cell membrane is a lot of different pieces coming together. It's not just phospholipids. There are other things embedded in that membrane. So let's take a look at some of those. These components can be lipids, carbohydrates, and protein. And one lipid that we've already discussed that is found in the cell membrane is cholesterol. Now, we spoke previously that fluidity can come from space between the phospholipids, and cholesterol basically is a space filler. It adds space to the cell membrane, and this reduces fluidity. So we don't just find phospholipids, we find cholesterol molecules, these linked carbon rings embedded in the phospholipid bilayer where there would be extra space so that there's not as much movement, there's not as much fluidity. So it increases stability, reduces fluidity. Now there's a second thing that cholesterol does, and this is sort of a temperature, it reduces the effect of temperature. By increasing the rigidity, it allows the membrane to withstand a smidge of higher temperatures without melting. It kind of keeps it together a little bit longer. And it, because it's kind of a, because it's a space filler, it actually prevents the membrane from freezing for a smidge. So when temperatures get really cold, they can't, the phospholipids can't lock together in this like solid crystal structure that happens with solids, because there's cholesterol in the way. It takes it longer. There has to be less temperature for it to actually get to a solid state. So it actually increases fluidity a little bit at cold temperatures, but for the most part, it decreases fluidity in general. So thank you, cholesterol. Now, there are lots of proteins that get embedded in the cell membrane as part of this mosaic component. And in this model, they're very cartoony, but the proteins can have a lot of different functions. They can be receptors that attract specific material to be enveloped in a vesicle for receptor-mediated endocytosis. They can be recognition proteins, because sometimes cells actually recognize and interact with each other. So I know we've talked about recognition proteins as determining destinations for vesicles, but they can also have one cell type recognize another cell type, and that type of interaction is quite important just for regular physiology, but also the immune system. You know, we have certain cells that attack and identify other cells. Well, there has to be recognition proteins for them to do that, and antibodies get involved, and that's a whole other unit. But recognition proteins can be embedded in the membrane. We also have transport proteins, which we've seen a few videos discussed briefly, that can, like for instance over here, we have this channel that's open that lets maybe water pass through it, or we can have the sodium ion pump, or we could have the glucose sodium pump, which allows specific molecules in due to the polar changing of the enzymes. And we can have other enzymes whose job it is to either synthesize or digest something right there in the cell membrane. So all of those can be embedded. Now, this model kind of gets the idea that, hey, there's a whole lot of different types of stuff that can be there in the cell membrane, but it's inaccurate because it's very cartoony. So here we have a slightly more accurate model where we can see that these proteins are this long polypeptide chain that have secondary structures such as alpha helices. So we've got the alpha helices here, and it also kind of includes carbohydrates. We can have proteins that have carbohydrates attached to the proteins. These are called glycoproteins, and they can serve functions for cell signaling or cell recognition in addition to just the regular proteins. So we've got lipids involved with the cholesterol, we've got carbohydrates involved with glycoproteins, we've got a whole slew of regular proteins that can be embedded. And finally, this last model is the most accurate because it's a big mess, which is how proteins really are and how these are. So you can kind of tell that this is our phospholipid bilayer. We've got the phosphate heads over here with this like orange reddish color, and we've got the green, fatty acid tails, and that's mirrored on this other side. And then inside we have all these protein subunits with their alpha helices, and the arrows represent beta sheets and these parallel lines bound to each other, and it's just this big mess. So the cell membrane is a big mess. That is the fluid mosaic model. I hope this helps. Struggle well.